we are looking at retroviruses and HIV AIDS in particular. Are you able to see my slides? And can you hear me clearly?
Yes.
Okay, thank you. So, yeah. So in this lesson, we are going to talk about the distribution of retroviruses and HIV, and the characteristics and pathogenesis of the virus, and the clinical manifestations, and the drugs and the prevention strategies that we have in the field. And this lesson has a lot of crosstalk with the first lesson that we had around 2 weeks ago on the immunology of two viruses, because there we used HIV as an example. So I think things will not be new to you. And just to emphasize the importance of studying HIV and understanding how to deal with it. As at now, we have around 38 million people who are living with HIV in the world as at 2021, based on the estimates. And in fact, since the time when the HIV outbreak happened, we have had around 84 million people being infected from the beginning, and around 40 million people have died. And we still have new infections that are happening. For instance, in the course of last year, it is estimated that around 1.5 million people were newly infected with the virus, and around 650,000 people died from AIDS-related illnesses. And of the 38 million that are infected, we have around 28 million who are accessing antiretroviral therapy, meaning that we still have around 10 million people who are not on any treatment, despite the scientific progress that we have had so far. So just to show you the reason why actually HIV is significant for us, in particular in the African setting, sub-Saharan African setting. Most of the HIV infections are actually in sub-Saharan Africa. So you can see that this region called Eastern and Southern Africa, of which Kenya, Ethiopia, other East African countries are, as well as all the Southern African and some Central African countries are. That region carries the biggest burden of HIV infections. When you look at the numbers, like last year, it was estimated to be around 20.6 million people in that region who are infected. Compared to other regions, you can see a little bit of infections in West Africa with around 5 million people, West and Central Africa region. So basically, you can see that sub-Saharan African region is carrying almost 25 million infections. Compared to other regions, where you find, like in Latin America, only 2.2 million people, in Northern America, where it's around 2.3 million people, including Western Europe, basically the West, and Eastern Europe, including Russia, with around 1.8 million. So you can see that the burden of HIV is heaviest in sub-Saharan African region. So that's the reason why we really need to understand HIV, because it's a fairly unique problem that we have. So you can see how heterogeneous the distribution is. And actually, even within these regions, you still have a lot of heterogeneity in distribution of HIV. So this is yeah, these are estimates that were reported in 2014 for Kenya. And when you use this key to try and estimate the prevalence in various regions, you can see that there are some regions like in Northeastern where the prevalence is fairly low. Like in Wajir at that time, it was estimated to be less than 1%. While you have some other regions where the prevalence is fairly high, above 5% for quite a number of regions like Nairobi, Machakos, Turkana, the majority of places. While there are some hotspots like in Nyanza, where some places you get even more than 15% of HIV prevalence. And but the national prevalence as at that time was around 6%, which hasn't really changed much. So it's very easy to look at the 6% national prevalence and then forget that we have hotspots and that probably require more targeted interventions. So we can look at HIV hotspots with regard to geography, but we also have some subsets of our populations that are also more affected by HIV. And these include populations like sex workers, people who inject drugs, and men who have sex with men. So you can see that with our national prevalence being around 5 to 6 or thereabouts, oscillating around that percentage, you can see that these key populations have very high prevalences of HIV infection, and they also have high incidence even as we speak now. And these key populations, in as much as they might seem to be small proportions of our national population, they actually account for 2%, for a third, 30% of the HIV infections. That they are 2%. So you can see that they are major drivers of the HIV transmissions in our communities. And this is one of the questions that has always been of interest to mankind. Where did HIV come from? And I know there are many theories out there, but the scientifically accepted theory of where HIV came from has been determined by comparing HIV with other viruses that exist that are similar. And we have very closely related viruses that we call simian immunodeficiency viruses. And some very elegant studies have been done in the wild to look at the natural hosts of those simian immunodeficiency viruses. People have been able to follow groups of these other animals in the wild and collected fecal samples and tested for genetic material that could be from SIV and compared the genetic sequences of those viruses with the ones that are seen in the human population as HIV. And so what has consistently come out is that the HIV that we know, the majority of HIVs that we have belong to a group called Group M. And this is showing a genetic tree, showing the relatedness of the various types of HIVs and SIVs from the animals. And the relatedness, the related viruses will cluster together, will be in closer branches. They'll be in closer clusters together. So what has been coming out from these phylogenetic trees suggests that the virus that we know as HIV, the HIV-1. So in HIV, we have two types of HIV. We have one called HIV-1 and another one called HIV-2. So most of the HIV viruses that are causing a menace in the community are HIV-1s. And what has come out is that those viruses closely resemble the viruses that have been isolated from chimpanzees. the SIV viruses isolated from chimpanzees. And so that has been attributed to the main group of HIVs, which we call group M, and also to other groups like group N. You can see that they are intermingling with the SIV chimpanzees, which are shown here and here. So you can see those branches are closely intermingling. Recently, we have also seen that gorillas also have very closely related SIVs that are very closely related to the HIV-1, but they seem to cluster more with group O. So suggesting that gorillas, the HIV that we have in humans that is classified as group O might have originated from gorillas. And whether the gorillas got it from chimpanzees or whether the two actually had similar viruses, we don't know, but it seems to be more closely related to the one in gorillas. Then we have HIV-2 in humans, which is mainly in West Africa, and it seems to cluster very closely with an SIV from a monkey that is common in West Africa called the sooty mangabe. And so it has been assumed that the HIV-2 crossed over from the sooty mangabes to humans in West Africa. And it's actually quite interesting that the geographical distribution of these animals also mirrors the distribution of HIV in the human population. So that is even stronger evidence suggesting that these are the monkeys that might have acted as the origins of HIV for it to cross over into humans. And actually, most of the monkeys out there have various forms of SIV. The majority of monkeys in Africa, actually, I think even all of them, they have various forms of SIV. And the only difference is that they don't usually get disease progression because they have co-evolved with HIV for so many years. Therefore, the monkeys have been selected for the ones that are able to coexist with HIV, with SIV, and the SIV has also been selected for the variants that are able to coexist with their host, because I mean, viruses will want their host to live long so that the virus can also continue replicating and transmitting. So the majority of the monkeys are able to coexist. For instance, the African green monkeys, which are common here, they are able to coexist with their variants of SIV without ever developing disease because of that coevolution between the host and the virus over many generations. But the fact that HIV probably crossed over from these animals to humans in the recent past, the scientific community thinks that that's the reason why HIV is able to cause disease in humans and lead to death, because we have not co-evolved with it as humans. So that is the idea. And then you could ask, how did it cross over from these chimpanzees, for instance? There's a lot of close interaction between these monkeys and these chimpanzees, and basically there's very close interaction in some of the regions between these animals and humans, where these animals are hunted for game meat. So, and in fact, in some investigations, it was found that a lot of game meat from monkeys in Central African countries had one form of SIV or the other form of SIV, suggesting that in as much as humans are not generally infectable with these SIVs, there's continued exposure, and therefore there's always the risk of crossing over when humans are handling these meat products. And also some of these monkeys are kept as pets, so there's also very close interaction but mainly from gamete. So those are the hypotheses that are there. I know there are many other hypotheses out there, but those are the scientific hypotheses that do exist. And some of the scientific backing for those hypotheses is that some studies have been done by looking at all the HIV viruses that we have sequenced in humans. If you are able to look at the sequences and know when this sequence was isolated, you'll not, for HIV, you'll not really know when it was transmitted, of course, but it will give you a rough, it can give you a rough estimate of when it might have been transmitted, even if it's a decade, you have a span of a decade. And then if you look at those sequences, you can use some analysis that we call analyzing molecular clocks. You can look at the mutations that have been accumulated over time. And because you are able to estimate the frequency of mutations over time, you can then work backwards to when the common ancestor of all those viruses can be traced, when it can be estimated that there was a common ancestor of all the viruses that you're analyzing. And so that's the reason why it has been possible for people to estimate when the SIV from Sutimanga base could have mutated and crossed over to cause HIV2 at around 1930s and HIV at around early 1900s. So that's the scientific evidence that we have, and it has been a big debate. It will be interesting to hear what you say after this discussion. So all retroviruses belong to this family, Retroviridae, as the name suggests, and within that you have subfamilies, mainly 2 subfamilies, and then you also have the genera and HIV. HIV1 and HIV2, and also the SIVs that you are talking about belong to the lentivirus genera. And you also have some others that also happen in other animal models, in other animals like feline immunodeficiency virus, bovine immunodeficiency virus, and then this one which is in pigs, equine immunodeficiency anemia virus. So those are the ones that are of most interest today. So retroviruses, they undergo reverse transcription. And what is reverse transcription? Anyone who has an idea what reverse transcription is? Perfect. Yeah, converting RNA to DNA. Because ordinary transcription is converting, transcribing DNA to create RNA. So these are the viruses that are able to do reverse transcription, and as the name also suggests. And all the retroviruses have 3 main genes, GAG gene, POL gene, and ENF gene. We are going to look at the structure, and yeah, and they also have 2 copies of positive sense single-stranded RNA. And in the previous class, I think, oh yeah, I forgot to answer someone about the Baltimore classification. So for HIV, it's group 6 because of the single-stranded RNA and reverse transcription nature. And this is the International Committee on Taxonomy of Viruses, the classification, family Retroviridae, genus lentivirus, and then you have those 2 species that we have talked about. Oh yeah, and you might have wondered when earlier on I was talking about groups. So like in HIV1, we have groups within that, group M, N, O, P, and these groups are thought to indicate independent crossovers from chimpanzees. So, and because when you look at them, they seem to have different clusters, they form different genetic clusters. So this suggests that HIV has been able to cross over, HIV1 has been able to cross over from chimpanzees several times that are known so far. And this just shows how at the end of the day, we ended up having HIV-1, because it means that transmission crossing over is something that can happen again and again. And it means that it might have also been happening previously, only that wherever it crossed over, it was not able to spread to reach to spread to larger populations. Yeah. And then within group M, we have many clades, and these clades are thought to have evolved now within humans separately. So we have clade A, K, A all the way to K. In Kenya, we mainly have clade A and D, and also we have some forms that are called circulating recombinant forms. In the southern part of Africa, we have clade C, and then in the west, most of the infections are clade B. In a place like Uganda, we have a lot of clade D. And then also HIV-2 also has some clades, which are basically just subgroups based on how the virus evolved. So this is meant to simplify all that information. So we have HIV-1 and HIV-2. So we are going to dwell more on HIV-1. HIV-2, as I mentioned, also has clades. But in HIV-1, I talked about group M, N, O, P. Remember, group O is the one that has been suggested to have originated from gorillas. Group N, group M, from chimpanzees, and then I can't remember about group P. We can just have a look at the phylogenetic tree here. Yeah, so you can see group O is very closely related to the gorillas, and also group P seems to be also very closely related to the gorilla SIV, while group N and group M are very closely related to the chimpanzee SIV. And then so within group M, M is for major because it's the main one. That's where you have all these other clades, and the majority of HIVs in the world are caused by group M. You have all the way from group A all the way to K, and then the circulating recombinant forms, where you find a virus which has a portion which looks like group C, and another portion which looks like clade C, and another one which looks like clade G, for instance. So HIV-2 is less studied because it is also less problematic, and it's mainly restricted to West Africa and a little bit in Europe and USA, while HIV-1 is the one that is distributed worldwide and is more easily transmissible and also leads to faster progression. In fact, the majority of HIV-2s are thought to be not progressive, and therefore people end up being like what we call long-term non-progressors if they have HIV-2. And then, as we said earlier, the SIV from chimpanzees is thought to be the main source of HIV that is in humans. Remember, chimpanzees are the ones that we are most closely related to in the animal kingdom. And so no wonder it was easier for HIV to cross over from chimpanzees. And then group HIV-2, we earlier mentioned that it is thought to have come from sooty mangabeys. And then we have a wide range of treatments for HIV-1. We have, one of them is just, this is just one of the treatments for HIV-1. Non-nucleoside reverse transcriptase inhibitors. We also have nucleotide reverse transcriptase inhibitors and protease inhibitors. We have many others. But what is interesting is that HIV-2 does not respond to this class of drugs, but it does respond to the others. So this is how the HIV virion looks like. So this is an enveloped virus, meaning that when it is being produced by the infected cell, when the infected cell is churning out new virus particles, they usually live with a portion of the cell membrane, and that's what gives it an envelope. And that envelope usually has what we call envelope glycoproteins, and GP120 is one of them, and then the other one is GP41, and these ones are usually in trimers. And these are the machinery that HIV uses to attach to target cells. And we talked about these ones in the class that we had on viral infection, immunology to viral infections. And these are the ones that we usually try to target with antibodies so as to neutralize the virus. But then we also have other structural proteins, like we have the P24 protein, which forms some of the internal structures. We have the P17, which forms also internal structures. In addition to that, we have enzymes. So the virus usually carries some enzymes with it, which are important for the transmission and infection of the target cell once the virus has been able to penetrate a target cell, like integrase, reverse transcriptase, and it also has what we call the nucleocapsid, which are also part of the structure, the internal structure. And then it has what we call accessory proteins, like VIF, VPR, NEF, P7. So these accessory proteins are quite important for the virus because they enable it to either replicate more efficiently. There's another one called TAT, which is actually quite important for viral replication. And then some of these like VIF, VPR, NEF, they are also important for immune evasion. They dampen the immune response and enable the virus to have more efficient infections. Protease is one of the enzymes on top of the reverse transcriptase and integrase, and that is important for the maturation of the particle. So as we mentioned in the previous slide, the machinery by which HIV attaches onto target cells is the surface glycoproteins, the envelope glycoproteins, of which GP120 and GP41 are part of. And it usually targets cells that express CD4 and also co-receptors. CD4 receptor is the main target receptor, but it also has co-receptors, which are mainly what we call chemokine receptors, and we have two, CCR5 and CXCR4, which the envelope glycoproteins are able to recognize. So it needs both of them, and these are receptors that are mainly found in immune cells, like CD4 T cells, some T cells, which are the CD4 T cells, and also some antigen-presenting cells like monocytes and macrophages. So just a very brief coverage on the replication cycle. So if you have a virion, which is a free virion, it will look for a cell that has a CD4 receptor and a receptive chemokine receptor, like CXCR4 or CCR5, and then it will come and attach onto that target cell. Then it will undergo what we call fusion, whereby it's basically entry of the enzymes and the genetic material and some of the internal material from the virus particle are released into the target cell. And then now, so that means that you have viral RNA inside the cell, and you have the enzymes like reverse transcriptase inside the target cell. So that is able to mediate reverse transcription, and then you generate viral DNA. And this viral DNA then forms what we call a pre-integration complex, because the main target of this viral DNA is for it to go and get integrated within the host genome. And so it will form that complex which goes into the nucleus and inserts itself within the DNA of the host cell. And then from there now it takes over the cell machinery for protein expression. And that's how, and for transcription to generate new RNA, and then that RNA can then, some of it will be genetic RNA for the virus itself. Then you will also have, which here we are calling the viral RNA, while some of the mRNA, some of the RNA that will be produced will be mRNA that will be used for translating the other proteins that are required to form the virus particle. And so those virus proteins are then translated. So you have transcription from the integrated DNA, which is called the provirus. into mRNA, and then you have translation into proteins. And then on the other hand, you have viral RNA being produced, and then they assemble together. And then some of those viral proteins, like the glycoproteins, the envelope glycoproteins will be inserted onto the host cell membrane. So when the virus is forming next to the cell membrane, it will bud out with a piece of that cell membrane, and that will be the envelope of the virus, which will also have the envelope glycoproteins. And then you have the virus particle. But initially, when it is budding out, it is an immature virus. And then one of the proteins that will have been produced here is protease, the protease protein, which can cleave some of the proteins further in further maturation. And then you have an infectious virus that can go and do the same process in another cell, infect another cell. So most of the transmission of HIV is through sexual contact, and this can be mainly anal and vaginal. You can also have oral transmission, but rarely. But if there are breaches in the mucosa, but most of the transmissions of HIV will be through vaginal and anal sex. And then you also have the transmission that is through needles in people who are injecting drugs or anything that is associated with the exchange of blood could lead to exposure to HIV. And then blood transfusion, if the blood is not screened before transfusion. And then you also have mother-to-child transmission during pregnancy, or also sometimes during delivery or even in breastfeeding. So basically, exposure to infected body fluids is generally how HIV is transmitted. So what makes some exposures infectious and others might escape infection? Because transmission of HIV is not always happening wherever someone is exposed to HIV, but the risk can be increased in some cases where someone is exposed to very high doses of HIV, or if the infecting partner has very high viral loads, of course the risk of transmission is higher. And that's the reason why nowadays we have a policy of when someone is diagnosed with HIV, they are treated immediately, which is good for the immune system because the integrity of the immune system will be maintained. But also it also reduces onwards transmission to other people. And then we know for sure that some hosts are less susceptible to infection. If someone has a mutation in one of those core receptors, like the CCR5, we have a proportion of people, especially of Caucasian descent, who have a mutation in the CCR5 gene, and that makes them less receptive for infection with HIV. So, and we'll come back to that at the end when you're talking about how some of the treatments for HIV have been done. And also, some routes of transmission are more have a higher risk of transmission, like intravenous has higher risk than sexual transmission, and for sexual transmission, rectal transmission has a higher risk compared to vaginal transmission. And this is mainly driven by the biology, the human biology, whereby the vaginal route is more adapted for having a better barrier. And then also sometimes when people have co-infections, like if someone has STDs, because of that breach of the mucosal barrier in the genital tract, they are more likely to get infected. So we talked about the replication cycle at the cellular level, but what happens to the patient, to an individual who is exposed to HIV? If the transmission is successful, the first cells that will be infected are cells that express CD4. This is not necessarily CD4 T cells, but it's just cells in the genital mucosa that do express CD4 and the core receptors. And This includes some of the antigen-presenting cells, like some of the kinds of macrophages, but just generally cells that are receptive to the infection. And then once that happens, it means that then those cells have a chance to spread the infection to other neighboring cells in the genital tract that are also susceptible. And then eventually you have dissemination to the general circulation, where then you'll be able to detect the virus in blood. And then later on, we talked about this in the other lecture 2 weeks ago. You'll have immune responses checking in to somehow control the viral replication partially. So the person is able to clear most of the virus, but some of it persists, and in what we call the viral set point, and that continues causing some destruction of the immune system in the background. So we usually divide HIV infection into 3 stages generally. In some cases, it is divided into 4 stages, but generally in 3 stages. Where you have the primary and acute HIV, which is just a few weeks after exposure. And this is the time when someone has not yet started making immune responses. That's when this phase starts. So you find that initially there's viral replication that is not checked by immune responses, and also later on when the immune responses check in, you have people seroconverting. If you look for antibodies against HIV, at some point you start being able to detect them in those individuals. And during this time, because these are new viruses, the body has never seen it, and it is uncontrolled by the immune system, people end up getting what we call the retroviral syndrome, acute retroviral syndrome, which presents as a flu-like illness and generalized swelling of lymph nodes, lymphadenopathy. So that happens during the first few days, but then the body is able to control the infection after a few weeks, though not fully, but to a certain level. And then that's when people go into what we call the asymptomatic stage, which can last even up to 10 years. But damage to the immune system will be happening slowly in the background without being noticed. And then at the end of the day, when the immune system has been damaged fully, it allows emergence of opportunistic infections and terminal stage disease, which is what we call AIDS. So this is a summary of that information. You have the acute phase, acute and early HIV infection, when you have viral loads, as shown in red, skyrocketing to a peak, and then the immune system starts checking in at some point and controls the viral replication to some level. And then there's a viral load set point, which is maintained with the gradual changes over time. And this is clinical latency period. And then later on, the immune system gives in, and you have opportunistic infections emerging. And you can see the kind of dynamics that happen on the immune system. If we use the CD4 counts are a good proxy measure of the integrity of the immune system. You can see that during acute infection, there's a sharp decline, partial recovery when the immune system checks in to control the virus, and then some decline in the background without notice, up to a point where the immune system just gives in and the virus replication skyrockets again. So some of the opportunistic infections that will be coming up at the later stages are things like herpes zoster, giving what we call shingles. These are viruses that in someone who has a good immune system, they will remain suppressed. They will remain there, but they will be suppressed by the immune system. But when someone is immunosuppressed by HIV or any other thing that can cause immune suppression, you have these kinds of manifestations. So just additional opportunistic infections, in addition to that, in addition to the herpes zoster, the shingles, You can have later on when the AIDS stage starts, you can have even protozoa infections that are generally controlled by the immune system adequately, like toxoplasmosis, cryptosporidiosis, isosporidiosis. All of these are protozoa infections that ordinarily would not cause a lot of problems in someone whose immune system is good. Things like candida infections, like oral thrush, they just become a common infection in such an individual. Cryptococcal meningitis, histoplasmosis, all these fungal infections start also gaining traction in these people at those late stages. Things like TB, even though actually TB is also, the chances of acquiring TB even during the latent stages are still higher in people who are HIV infected, even in during the latency stages, even during this clinical latency period. But it becomes a bigger problem at those end stage disease stages. Just general bacterial infections become a menace. And then, as we saw earlier, things like varicella zoster virus, shingles, such as things like shingles. If someone had some suppressed herpes infection, they start manifesting. CMV, cytomegalovirus, they start manifesting. So it just becomes a big issue where the immune system is unable to control all these infections, and then they start cropping up. So you can classify these ones based on where they manifest, those opportunistic infections. You have some that you'll see on the skin, as we saw in the earlier photos, things like herpes viruses, shingles. Also, we have some AIDS-related tumors, cancers like Kaposi sarcoma, which will usually manifest on the skin and also on mucosal surfaces. Then you have the ones that will cause respiratory complications like TB, pneumocystis, pneumonia, also things like Kaposi sarcoma because you have mucosal tissues there, bacteria. Then the GIT, you have things that will cause a lot of diarrhea. And then in the CNS, you have some of these viruses causing CNS manifestations like encephalitis, or even some of the protozoa also taking hold and causing serious problems in the CNS. Also some lymphomas, non-Hodgkin lymphomas that will also cause CNS manifestations. So earlier on, I had not talked about tumors. So these are some of the tumors that are associated with the AIDS and generally HIV infection, like especially uncontrolled HIV infection, which has progressed to the end stage. Things like Kaposi sarcoma, lymphomas, and also we also noticed that people who are HIV infected and they have HPV infections, they have a higher likelihood of developing cervical cancers compared to people who are not HIV infected. So this is not necessarily only associated with the HIV patients. I mean, even people who are not HIV infected, as we saw earlier, could develop cervical cancer if they are infected with the high-risk HPV types. But it's just that we tend to see cervical cancers more often in people who are HIV infected when you compare with people who are not HIV infected. So this showing Kaposi sarcoma. The name is a bit of a misnomer. It's not a sarcoma. Actually, it's a cancer of the epithelial cells in the lymphatic vessels. But at the end of the day, it ends up manifesting like this because you have lymphatic vessels anywhere. More of Kaposi sarcoma manifestation in the mucosal tissues. You can see oral thrush in someone who has a suppressed immune system, and most likely an HIV patient. Something that is fairly uncommon in other people, except when they have some form of immune suppression. So let's look at HIV testing. So we do HIV testing first to diagnose, to see whether someone is HIV infected, so that we can initiate treatment, because we have fairly good treatments. And also, we also do it for surveillance to know how much HIV is circulating in our community, and that will guide us in making appropriate policies to control HIV transmission, and also to provide treatment to people who are HIV infected. So that's mainly for policy. And also when blood and organ transfusion, because HIV can be easily transmitted through blood and organ transplant, blood transfusion and organ transplant, we usually do HIV testing also. And we still have a few countries that usually require someone to test for HIV. And in fact, in some countries, if you are a foreigner and you are HIV infected, you could be deported back. In other countries, there will be restrictions for traveling if someone is HIV infected, even up as at this day. But the majority of the countries have removed that ban. And for the HIV tests that we have, we can classify them into 3. I know some of you will probably add another one, the one you call kupima kwa macho. So yeah, but basically the test that we use is we have the antibody test, antigen test, and nucleic acid test. Of course, you don't expect someone to have antibodies against HIV if they are not infected. So if we measure antibodies, we'll be able to determine if someone has HIV infection. So amongst those tests, we can divide them based on whether they are serological tests or nucleic acid tests. So serological tests are the ones that you do in blood, on serum. And so I talked about antibody tests, antigen tests, and nucleic acid tests. So amongst the antibody tests, we have what we call rapid HIV tests. And we also have an ELISAs that are able to measure antibodies in blood. And we also have what we call Western blot analysis. These were the ones that were used at the very beginning in the 80s. In fact, at the beginning, they were the only ones that were available, and then the others came along the way. And so all these are mainly used to measure antibodies in blood. And the assumption is if someone has antibodies against HIV, it means that they are responding to an HIV infection that they have acquired. Some of these tests, like the ELISA, has also been modified recently, so that it measures for both antibodies and antigen. So we call it an ELISA combi test, because it's a combination of both antigen and antibody test. And in fact, also some of the rapid tests are also, the most recent ones have also incorporated antigen testing, but they are mainly skewed towards antibody testing. And then we have an antigen test, which has been the mainstay for antigen testing, which we call the P24 antigen. Remember, P24 is one of the proteins that are in the HIV particle. So you are able to actually measure and try and quantify if someone's blood has the P24 antigen. And in that way, you are not actually measuring for the immune response, but you're actually trying to detect the virus proteins themselves. So yeah, there's something else I wanted to add here. So we have said that the ELISA test and the rapid tests also have incorporated some reagents within them where they are measuring both antigen and antibody to some level, but the P24 antigen test is specifically for determining the antigen. But now, the P24 antigen only detects antigen during early infections before antibodies develop. Because what usually happens is that once the immune response develops and antibodies develop, all the free P24 antigen that is there will get coated with antibodies, and then it becomes difficult to detect the P24 antigen later on. But in any case, at that time, you're able to measure antibodies, so you'll still be able to to diagnose HIV infection using the other tests. So this is an example of a rapid test. We use this quite often, the Unigold test, where you are able to differentiate a positive test from a negative test based on whether the test band is positive. You just put a little bit of blood here and some buffer to drive the sample through the system, and then within 10 minutes, you are able to diagnose HIV infection. And this is an example of the ELISA tests that we do in the lab, which are more sensitive than the rapid tests, and that's the reason why we usually use them for confirmatory testing. And then, yeah, and this one also has, this one is the one that has a combination. Initially, it was for antibodies, but it has now a combination of both measuring antigen and antibody. And yeah, we have the nucleic acid tests, which is the other category. Remember here, we have all along talked about serological tests, which are mainly the first two, which mainly accommodate the first two kinds of tests, the antibody and antigen tests. But then we have the nucleic acid test. So here we usually measure for the viral genome through polymerase chain reaction. And we can do it both quantitatively or qualitatively. Qualitatively is where you just want to detect whether this person has a genome, viral genome in their blood. And quantitative is where you actually want to quantify. And we use the quantitative test to also monitor viral loads over time, so as to see how people are responding to treatments. And we also use this PCR test to test for HIV infection in infants, in infants generally. And this is usually when we are following infants whose mothers are HIV infected. So why do you think we use this one for monitoring HIV acquisition in infants? Any ideas? Why don't we use the antibody tests to try and check for HIV infection in infants? Someone said immature immune system. Another one said early detection. Child immunity is slow. Have you? It's faster. No, it's not faster, and it's not because of the immature immune system. Yeah, one person gave a very correct answer. Infants get antibodies from the mother through transplacental acquisition of antibodies. So when a mother is infected with HIV, they have antibodies against HIV, isn't it? They are able to pass those antibodies through the placenta to the child before birth. So that means that that child, if you take a rapid test or any antibody-based test, they look as if they are HIV infected because they have maternal antibodies. So the only sure way you can determine if a child is actually HIV infected is by measuring the virus itself, by monitoring the viral genome by PCR. So we have various drugs that we use to treat HIV. Some of these drugs were available as early as late 80s, and we have had many others that have been developed along the way. So we have the nucleoside reverse transcriptase inhibitors. Basically, these ones, they mimic nucleosides and therefore prevent the generation of genomic material for the virus. So that's how they are able to curtail viral replication. We also have the non-nucleoside reverse transcriptase inhibitors. And so basically, the non-nucleoside reverse transcriptase inhibitors also prevent production of the genetic material. And these two actually happen at the reverse transcription stage. So meaning that even if someone is HIV infected, in as much as the cells that are already infected will remain infected because the virus is already integrated into the DNA of those particular cells, there'll be minimal onward transmission of the virus to other cells, minimal replication, minimal establishment of the replication in other cells. So that's how they are able to curtail viral replication in the body. So then the nucleoside reverse transcriptase inhibitors just bind to the reverse transcriptase enzyme and prevent its function, and therefore that reverse transcriptase step is curtailed. Then you have what we call protease inhibitors. If you remember the replication cycle, we said that these viruses, when they are budding off from a cell, if the cell that's churning out viruses, because it's infected by itself, when it's churning out those viruses are actually immature. And then we have some enzymes that go and cleave some of those proteins that are immature so that the virus can become infectious. So protease inhibitors usually go and inhibit those enzymes, which are called the protease enzymes, to prevent the maturation of the viruses that are being released. And that's how viral replication is curtailed. And we have quite a number of those protease inhibitors. And then we have others called integrase inhibitors. If you remember that replication cycle, once the viral RNA is reverse transcribed to DNA, it usually needs to enter the nucleus and go and integrate inside within the host cell's genome. And so these integrase inhibitors are able to prevent that process. And that means that the virus is not able to form proviruses. It is not able to integrate, and therefore that cycle is interrupted. And we have others which are only recently available, like Retroviruses, which is an attachment inhibitor. Remember, the virus, for it to infect a target cell within the body, it needs to attach to CD4 and a chemokine co-receptor. So Retroviruses blocks one of the co-receptors, the CCR5 co-receptor, and therefore prevents the cell from being infected because it's able to interrupt that interaction between CCR5 and the GP120. And then you have the fusion inhibitor. Once the virus attaches onto the target cell and has found a receptive CD4 and a receptive co-receptor, it usually undergoes some structural changes for it to penetrate, for it to release its internal material into the target cell. And so those changes that are happening at that point, when the virus is fusing with the target cell, they can be inhibited by some drugs, and one of them is the enfivitage. Then we have in addition to treatments, we do have other policies that are being implemented to try and control the pandemic. Currently, we have what we call 90-90-90 targets, where the aim is to have 90% of all HIV infections being recognized, being diagnosed, and 90% of them, of those people being put on ARVs, having ARVs available to them, and 90% of them, of those that are receiving ARVs, having viral suppression. And that means that they are able to live ordinary lives, and they are also less likely to transmit to other people, because one of the risk factors for transmitting to another person is having a high viral load. So that's one of the global policies that is being pushed. And then we also have the universal testing and treatment. Initially, we didn't have this. We have universal testing and then monitoring, and people would be given some antibiotics, cotrimoxazole, to minimize chances of developing other infections. But it was later on changed based on studies that suggested that immediate treatment was beneficial to the patient and was also limiting onward transmissions, somehow linked to the 90-90-90 policy. And of course, all this we want to implement this so as to eliminate the infections. Various countries are pushing for various prevention measures and control measures, which are now mainstream practice, like advocating for safe sex practices using condoms, and also screening blood products and organ donors before organ transplant. And also we have what we call prevention of mother-to-child transmission, where we have policies there where HIV-infected mothers who are pregnant are initiated on ARVs immediately. I mean, in fact, all pregnant women are tested to see whether they are HIV infected. And if someone is infected, of course, based on the diagnose and treat policy, they are put on ARVs immediately. And then also the child is also given ARVs to prevent acquisition of HIV. And early testing and linkage with care is something that we are pushing, and also the recognition that treating people prevents transmission of HIV to their partners. We have had some interesting studies that have shown that male circumcision, medical male circumcision, can prevent HIV acquisition, and therefore medical male circumcision has been also rolled out as a policy in communities where it is not routinely practiced. And then we also have pharmacological interventions that we use for preventing HIV acquisition. What we call post-exposure prophylaxis is if someone has been exposed to HIV, either sexually or from accidents, they are given ARVs to prevent acquisition of HIV, and that has been shown to be effective. And then we also have pre-exposure prophylaxis that has proven to be quite effective if taken daily, and whereby people who anticipate to have exposure to HIV from their partners, especially for disconnected couples, they are given ARVs to take daily. And those specific ARVs, in particular, Truvada is the one that is mostly used. Those particular ARVs prevent acquisition of HIV. And so for the PrEP medications, we are also having some long-term PrEP that has been shown to be effective, and that will be available in the clinic in the near future. We are also having antibodies, broadly neutralizing antibodies that have been developed and have been shown to also be able to prevent infections, acquisition of infections, and those might also, we don't have them being licensed in the clinic, but they might make it to the clinic in the near future. So, but our biggest issue is we don't have a vaccine yet to prevent HIV. We have had several vaccines that have been checked, and most of them have given disappointing results. But we have some new vaccine candidates that are in early phases of investigation, and it will be interesting to see how those will pan out. You might have heard about people who have been cured of HIV, and as far as I know, we have had only 3 people who have been cured of HIV, and we have the person we call the Berlin patient, and we also have another one called the London patient, and there's another case that was reported sometime just recently, fairly recently, the Chicago patient. And these are people who have received bone marrow transplant. If you remember earlier on, we talked about some people being resistant to acquisition of HIV because they have a mutation in the CCR5 gene, because HIV will require the CCR5 to be able to use it as a core receptor to enter target cells. So here we have had a few people who were HIV infected, and then their bone marrow was replaced with bone marrow from someone who has CCR5 mutation, a delta 32 CCR5 mutation. And that means that the new bone marrow these people had, after removing their previous bone marrow and deleting their previous immune system, they were able to create a new immune system from the transplanted bone marrow which is HIV resistant. And that's how these people were able to get cured. But then that's a very invasive procedure, fairly harsh. And therefore, these people have only received that bone marrow transplant because they needed a bone marrow transplant to treat various kinds of cancer, either leukemia or lymphomas that are usually treated by ablating the immune system and replacing it with a new bone marrow in a bone marrow transplant. So this is not the kind of thing that can be rolled out en masse, but it has only been used as a proof of principle that if someone is HIV infected and then they have a lymphoma that needs to be cured using a bone marrow transplant, that scenario can be used where you go and get a donor who is Delta 32 mutant, and therefore the recipient patient will become Delta 32. They'll have an immune system which has a Delta 32 mutation, and therefore HIV replication will be stopped. Okay, so that's it. Any questions? I can see there are many questions in the chat. Let me look at them. Hey, you guys have had a lot of discussions here. How did humans acquire HIV from chimpanzees? We talked about that. It's probably due to, no, it's unlikely to be from sex. It's probably due to exposure to blood products. Yeah, you'll find a lot of game meat from those animals being consumed in some of the countries. Oh, I can see someone talking about the polio vaccine hypothesis. Please just go and read around that, but there isn't much evidence about it. But it is one of the theories that have been floated out there about how HIV might have spread from primates. I'm just scrolling. Yeah, human has the option of eating cows, but just to eat gorillas, okay. Any question that you want to, if you want me to answer any question, because I can see it will take me long to scroll, just post the question at the bottom. Can you go over the structural and accessory proteins again? Okay, I'll go back to that. Yeah, so the structural proteins are the ones that are associated with making the, as the name suggests, the physical structure that is important for the integrity of that particle, the HIV particle. So you can see like the capsule proteins, the matrix proteins like the GAG P17. These are basically important for the physical integrity of that virus particle. The GP120 and the GP41. So those are the ones that we call the structural proteins. Then you have the enzymes. Enzymes will be things like integrase, reverse transcriptase, and protease. Those are the main the enzymes. Then you have accessory proteins, things like VIF, VPR, NEF. Those are the ones that are important for the virus to evade the immune system. NEF will usually downgrade, lower expression of HLA. If you remember the lecture that we had for immune responses to viruses, antigen presentation takes place through HLA molecules. So NEF will lower the HLA molecule expressions in the infected cells, and that's how it will prevent the immune system from recognizing that that is an infected cell. While things like, there's another one here which I have not included, called TAT. TAT is important for enhancing the transcription of the virus material from the provirus. And that's how it's able to enhance viral replication. So the accessory proteins are mainly enabling enhanced viral replication. They are basically enhancing viral replication and also enabling the virus to evade the immune system. Something like VIF is able to cause degradation of some proteins in the cell that usually prevent viral replication. Some proteins that are part of the innate immune system will be degraded because of presence of VIF. Okay, what is the difference between nucleoside and non-nucleoside reverse transcriptase inhibitors? The nucleoside ones, they look like nucleotides, and therefore they mimic something like cytidine. You know the three nucleotides, huh? ATGC, cytidine, guanine. So they'll mimic those ones, and therefore they'll be incorporated in that chain when the HIV genome is being transcribed. They'll be incorporated there, or when the reverse transcriptase is actually reverse transcribing the RNA to generate the DNA, they'll be incorporated there. But because they are modified, they will not allow the elongation of that DNA chain that is being produced from the reverse transcriptase, reverse transcription. And so you'll end up with immature an immature stopping of that elongation of that DNA. So that's for the nucleoside reverse transcriptase inhibitors. The non-nucleoside ones, they just bind to the reverse transcriptase enzyme, and they make it defective in its function, and therefore it will not reverse transcribe. By daily, you mean PrEPs are taken daily with or without sex for discordant couples, or just the days they are engaging? It is daily, because you need to, it takes a while for, when someone is taking PrEP, they usually do it daily, according to the PrEP that we have right now, because adequate levels have to be maintained. And it takes a while for those adequate levels to be attained, especially in the mucosal compartments where the exposure is taking place. So that has actually been the biggest issue with PrEP, because it means that people find it difficult to adhere to the medication by taking it every day. And sometimes people will just remember to take it because they had some sexual exposure. So that has been the biggest limitation with PrEP. Because if you just take it just before, you will not have had the required levels accumulating in the tissues, in the target tissues. But as I mentioned at the end, we have some PrEP formulations that are coming up, which are slow released, sustained release preparations that can enable someone to get an injection once a month or once every 2 months. How do we know the specific class of ARVs to give to a patient? Are we really able to specify and say this one needs a protease inhibitors and not an integrase inhibitors, or any just ones? So there are some very well-defined regimens, and this also differ from country to country. So the day you go to the clinic, you need to find which regimens are used. But generally, we have combinations because HIV is very mutable. It mutates very easily. So you cannot give someone just one drug. They are likely to escape. The virus is likely to escape, and then that drug will become ineffective. So we have what we call combination therapy, which is part of the clinical guidelines. So it will be important to refer to the clinical guidelines as at that time in the hospital that you're practicing in, so that you give the 3 drugs or even more, depending on the treatment guidelines as at that time. Historically, we have given 3 drugs which are in different categories, so that even if there's resistance to against one category, that is covered by the other 2 drugs. So I wouldn't want to give the specific combinations that we use, because they are always being updated. And also, when someone has resistance and they are moved to another regimen, that is less likely to have resistance, that they are less likely to have resistance to. Why is it that there is a higher risk of transmission in the vector? We think this is because of the physiology of the 2 organs, whereby the vaginal tract is more adapted to sex compared to the rectal region. But that is still not 100% understood, but that's what we think. Yeah, but the reality is that rectal transmissions are even tenfold higher compared to vaginal transmissions. If someone has the CD4 receptors but without either one of the co-receptors, will they still contract HIV? Those other co-receptors, someone will always have them. But if a cell doesn't have the co-receptors, that cell will not get infected. But I don't know of a cell in the body that has CD4 receptors but doesn't have the chemokine receptors. Those chemokine receptors are fairly expressed in almost all immune cells. A cell will have one or the other, depending on how the cell is meant to navigate its way around the body. So, but if a cell hypothetically doesn't have the co-receptors, it will not be infected by HIV. Why is weight important in ART, and why different regimen for different clients? So weight is important because you just want to give the correct dose. And the reason why people will have different regimens is because some people could be resistant to one HIV ARV, and therefore they might need a different regimen so that resistance is overcome. How do the doctors know that someone has that mutation? So ordinarily, people who are HIV infected will give out blood for testing viral loads when they are on treatment. And if you find that someone is still having high viral loads despite being on ARVs, it suggests that they probably have mutations that can make that virus resistant to the ARVs that this person is getting. So that means that there you will be able to suspect that there are mutations, but then to determine the exact mutations that the virus has, so as to guide you in selecting treatments, you'll have to do sequencing of that viral RNA to identify which mutations the virus has, and then you can choose the treatments based on that. Oh, you want to see my face? Someone says that I need to put my face. I will. I will do that. Which other? We don't have any HIV vaccine that has been authorized for use. We have had several phase 3. Phase 3 clinical trials are the ones where you are looking at whether the vaccine is effective in preventing acquisition of HIV. We have had several, but they have given disappointing results. We have the ones that we have that are in the pipeline are still phase 1, where we are actually testing for safety. Not yet. We are not yet at the level where we are able to test whether these vaccines are effective. Is it true that if you have been on long-term use of PEP or PrEP, you will develop ARV resistance once you get actually infected? This one, it's first of all, it's unlikely to get infected with good use of PEP or PrEP, unless someone is skipping dosages. Yeah, but if someone gets infected despite being on PEP or PrEP, it means that that virus is resistant to the drugs that that person is having. So that person, but that is a product of poor adherence, probably, to the drugs at the time when they were taking them for prevention. And yeah, so it means that the treatment regimen that is chosen after the person is infected would have to be different from what they were taking for PrEP. Yeah, the home test kits are basically rapid test kits. Some of them being the oral ones. They are basically just rapid test kits. Does the mutation in the CCR5 gene for the HIV-resistant individuals have other adverse immune effects? Not really. We don't see any. What happened is that these receptors, like the CCR5 receptor, they usually tend to have other receptors that are able to fill in that gap in the body. So there's a lot of redundancy for chemokine receptors. So that's the reason why people are able to remain okay. And in fact, there's that story that was going around about the Chinese scientists who did some gene editing for babies. And that's the receptor that they were mutating. What happens during HIV window period? That's when the virus is replicating in the mucosal compartments, in the tissues without it being known. And also, it also depends on how you define the window period, because we usually define it best as the period when you can't see antibodies. So it's also during the time when antibodies have not yet developed for you to allow the testing and detecting antibodies to diagnose. Yeah, if someone is diagnosed with HIV infection, regardless of their CD4 counts nowadays, someone is started on treatment immediately. So we have the test and treat. T-cell activation can increase the chances of acquiring HIV. And that's the reason why we also think having co-infections actually increases the chance of acquiring HIV, especially STIs. If someone has STIs, it means that they are also causing a lot of immune cell activation in the genital tract. Okay. So I will stop at that point. Because I understand there are some students who need to go for to the mosque. So I think we have had a good discussion, and see you in the next lesson. I don't know which is the next class that we are taking with you, but thanks for being very attentive and being very engaged. Njoroge, you have a question?
Yes.
Yes, please go ahead.
The first question I have is, you mentioned something to do with the way HIV infected people are administered with Septrin, and you talked about some studies which are saying that immediate treatment is better. Could you kindly elaborate on that issue?
Yeah. These studies have suggested that, okay, previously we used to defer treatment with ARVs, but over time it was shown that people who are started on treatment immediately fare better in the long run. In fact, they live ordinary lives. And when you look at the immune system, it seems to be intact. And that's the reason why it was changed, so that we don't defer treatments anymore, ARV treatments anymore, so that people are just initiated on treatment immediately. So at that time, people used to be just given Septrin if their CD4 counts are still high enough. That was good for preventing co-infections. There were stories that there were expectations that also this antibiotic, broad spectrum antibiotic, was also preventing general activation of the immune system, because it is preventing the pathogen load, the kinds of bacteria that the person is being exposed to. But basically at that time, the viral loads of the person would remain high because they are not on ARVs. And then we noticed that there was a lot of damage, not just to the CD4 counts, but also to other components of the immune system. So now the policy is, once someone is diagnosed with HIV, even if they were infected 2 weeks ago, as long as they are viremic and have been diagnosed with HIV, they are put on ARVs immediately. And that has been shown to lead to better longevity of these patients.
So because I realized the Septrin is still administered at the CCC pharmacies. So do they administer the Septrin together with the ARVs, or because I realized it is still there?
Yeah.
Oh, okay. Thank you. The second question.
It is still useful because because of that issue of being exposed to all kinds of bacterial infections.
Oh, okay. He also talks about voluntary medical male circumcision being a better preventing factor for HIV. So does that mean that some of our Kenyan communities, which are still using cultural methods of circumcision, are more at risk?
Not really. As long as the correct measures are taken to ensure that there's no exposure to HIV-infected instruments. As long as there's no cross-contamination of blood, there's no risk to that. So all that needs to be ensured is that there's no cross-contamination during those procedures. There are some communities in some parts of Africa where the traditional circumcision is actually quite different from what we call the medical circumcision. In most of the communities, it's the same. It's one and the same thing, because we remove the foreskin fully. And that's the part that the medical male circumcision will want to do, so as to minimize the mucosal surface that leads to exposure. But there are some communities where the transitional circumcision doesn't actually remove the entire foreskin. They just cut a small part of it. So that's the reason why we specify medical male circumcision, so that even if you are relying on the transitional one, it needs to be similar to that. Any other question?
Okay, the last question.
Yeah.
You talked about the opportunistic infections that arise when somebody is infected with HIV and AIDS, and maybe just to inquire, is it that whenever you have HIV and AIDS, of course it will depress the immune system, So does it mean that all the normal flora in the body, which are normally there at certain basal states without causing infection, will cause infection? For example, you gave an example of cryptococcal meningitis being one of the opportunistic infections, and yet Neisseria meningitidis is just a normal flora. So does that mean that all normal flora cause the infections or something?
No, not really. Actually, disruptions of the normal flora might be actually the big issue. Yeah. Some of those will not call them normal flora. Some of these bacterial exposures will be there, but they'll be suppressed because of the presence of the normal flora. So you cannot really say normal flora will cause infection. It is very specific ones that are capable of causing pathology, like the ones that were listed here. Any other question?
Okay, thank you very much.
Okay, because of time, let's leave it at that. Have a nice afternoon. Thanks, Joroge, for the questions.
Thank you.